Okay, let's just jump right in. We're doing a deep dive on a paper that really flips the script on how we think about treating genetic diseases.
It really does.
The problem it tackles is, well, it's mostly about logistics. You have over 200,000 known genetic mutations that cause disease.
Right.
And building a custom drug for every single one, it's impossible.
Financially, regulatorily, it's a non-starter.
So the big idea here is, what if you stop chasing every individual typo? What if instead you could upgrade the cell's own reading machinery just once?
And make it a permanent, durable fix for thousands of different diseases all at the same time. That's the core mission.
Exactly. And the specific target here is something called a nonsense mutation.
Yeah, these are mutations that create what are called premature termination codons, or PTCs. Basically, they're stop signs that show up way too early in a gene.
So the cell just stops building the protein halfway through.
And you get this truncated, useless protein. And this isn't some rare thing. We're talking about roughly 24% of all known pathogenic alleles in databases like ClinVar.
So a huge patient population.
A huge population. And for years, the dream solution was something called a suppressor tRNA. The idea was to have this molecule that would just insert an amino acid where that fake stop sign is, so the machinery could, read through it.
It never quite worked, did it?
It never worked. They were either way too weak to be therapeutic, or they were just too toxic, causing problems all over the cell.
Okay, I have to jump in here. 24% is a massive slice of the pie, I get it. But if this is a massive conceptual pivot, are we still just ignoring the other 76% of diseases?
Yeah.
The deletions, the missense mutations.
That's a totally fair point. And look, this isn't a silver bullet for every genetic disease. But for this class of diseases, it's a game changer.
Okay.
Think about it. Instead of thousands of different drugs, you could need just one therapeutic composition to treat them all. You're not fixing the mutation. You're fixing the consequence of the mutation.
You're solving the logistical bottleneck. That makes sense. So how do they do it? They used prime editing, and they called the system PERT.
Right. And their first step was logical. They had to find the best possible candidate, the best natural tRNA in the human genome to turn into this suppressor.
They didn't just guess.
No, they screened everything. All 418 human tRNAs. They systematically changed their anticodons to recognize one of the stop codons.
And they found a winner.
They did. The LuTAA tRNA family, it just had the best natural ability to do this job, the highest baseline read-through.
But baseline wasn't enough, I take it.
Oh, not even close. It yielded maybe 5% of the protein you'd need, so that led to the next step. Optimization. They took that promising chassis and, they fine-tuned the engine.
And they did this by introducing just 6 specific base pair changes. That seems surprisingly minimal.
Minimal tweak, massive payoff. That's the beauty of it. That fine-tuning boosted the performance like crazy. The optimized versions got up to 35% of wild-type protein yield.
35% from just a single copy in the genome.
Yep. That's a 5-fold increase over what they started with. A huge leap in power.
Okay, but this is where the big, scary question always comes up. The ghost in the machine for this whole field has always been safety.
Always.
If you permanently tell a cell to start ignoring stop signs, how do you know you're not just creating translational chaos, making it read right past the end of thousands of healthy genes?
That was the deal breaker, historically. The fear is reading through natural termination codons, the NTCs, the real necessary stop signs, but they tested for this directly.
With mass spectrometry.
Exactly. They looked for any evidence, any peptides at all, that would result from reading past those normal stop codons, and they found nothing. It was clean. It seems the cell have other safeguards, context around the stop codon decay pathways.
Yeah.
It's robust. The system didn't perturb the global proteome.
That is a massive safety checkmark. So that gives them the green light to actually try it on diseases.
Yes. So they took this single optimized PERT therapy and applied it to cell models for Batten disease, Tay-Sachs, Hurler syndrome, really diverse stuff.
It worked.
It worked. They saw anywhere from 17 to 70% restoration of enzyme activity. And in the Hurler syndrome mouse model, they restored enough enzyme activity to nearly completely resolve the disease pathology. We're talking about clearing out that destructive gunk in the brain cells.
Wow. So to zoom out then, the innovation here isn't just a new tool, it's a whole new strategy.
It's a profound conceptual shift. We're moving away from one mutation, one drug.
You upgrade a shared component. It's durable, it's disease-agnostic. They said they rescued over 70% of the specific stop codons they tested.
That's right. It's a platform, not a point solution.
So let's distill this for everyone listening. What does this all mean for you? I think there are three key takeaways. First, permanence is power. They use prime editing to install this fix permanently. It's a one-and-done treatment.
Unlike older transient approaches.
Second, this unlocks a massive patient population. It's a disease-agnostic therapy for, what, maybe a quarter of all inherited diseases.
The huge potential impact.
And third, the big one. The safety hurdle seems to be cleared. It restores the good protein without messing up all the others. So that leaves me with a final thought to mull over. If we can use genome editing to safely and permanently upgrade a fundamental piece of cellular machinery like this, where else can we apply this platform strategy to bypass the need for endless bespoke medicines?
Welcome to the Everyday Journal Club. I'm Hope, a biologist with 15 years of research experience, now fortunate to be working in a Nobel Prize-winning lab. This channel has three main goals. 1. To help you stay effortlessly updated on the latest scientific breakthroughs. No paywalls, no jargon overload. 2. To build a supportive community where we can exchange the most valuable experience-based research advice. 3. To share valuable strategies and concrete examples on how to achieve meaningful scientific breakthroughs. Once we reach 1,000 subscribers, I'll begin releasing content focused on the third goal. Let's go! Let's make great science together.